So let's jump right in with 17-3 and look at how RNA transcripts, or these primary transcripts that are made during transcription, can be processed in eukaryotic cells. Now, when thinking about this, to modify these initial RNA segments, what we do is we put some material on either ends of the molecule and we cut out some of the parts on the inside. So when you think back to the video that you saw in class, you have a molecule attached to, this is modified guanine, is attached to the end, the 5' end of the RNA segment, and that's called the 5' cap. And then on the other side, again, you saw this in the animation, you have what's called the poly A tail produced. And oddly enough, the name signifies the fact that there are a lot of these adenine nucleotides that are added to the end of the RNA transcript. Now, these particular additions have an important role because what they do is they help, the 5' end helps the RNA, messenger RNA, attach to the ribosome to begin translation, and the poly A tail helps prevent degradation or cutting up the RNA by enzymes. If there wasn't this protection on the end here, enzymes would chew up the coding segments of the RNA molecule. So again, this provides an area of protection. Let's see. So 5' end gets the 5' cap, obviously. 3' end gets the poly A tail. Again, helps protect digestion from enzymes and helps attach to the 5' end or 5' end attaches to the ribosome. Now let's take another look at this RNA that gets edited down. So we have our 5' cap and our poly A tail. Notice here in the RNA segment that you have lighter portions and darker portions. When after splicing occurs, notice the lighter portions are gone. These portions that get cut out are referred to as introns. Whoops, almost lunchtime. Introns are segments of RNA that actually get cut out and are never translated, so they don't code for any protein. Now, the way I remember this, I just think introns stay in the nucleus. Exons, on the other hand, are portions of this RNA that get spliced together or pieced together, and they will be expressed. I just remember that exons exit the nucleus and are expressed. They are used to make proteins. So exons exit and are expressed, introns stay in. So part of our editing of RNA has materials added to either end of the RNA segment, and then in the middle we cut out portions that are not used to make the protein. So this is just a summary of that. Introns, again, non-coding regions, they get cut out. Exons are expressed, they get stitched together. Splicing is the process by which this occurs, and I kid you not, this is done with an enzyme or a substance or complex, I guess is the best way to put it, called a spliceosome. Spliceosome, I mean, it sounds like something that somebody made up. But a spliceosome is basically these small nuclear ribonucleoproteins, they call them SNRPs, and some other associated proteins. But just think of it as the spliceosome. It does the splicing. Here's a way to visualize this. Here are these SNRPs and some proteins that join together. Now the SNRPs have these ends, these nucleotide ends, that can hydrogen bond with portions of the intron. So the SNRPs know where to attach, basically. And then the spliceosome will cut out the actual intron and then splice together the remaining exons. So if we look, there's exon 1, intron, exon 2. Spliceosome does its job and cuts out the intron and then stitches together the exons. That is the essence of this RNA editing. Now, some portions of RNA can actually catalyze their own splicing. Some portions of RNA can actually cut out their own portions of their own segments. These particular RNA molecules are referred to as ribozymes, again referring to sort of this enzymatic quality, or their catalytic quality, I should say. They're not actually enzymes. And what that does is provide evidence that enzymes are not the only catalysts that are possible. There are also these RNA catalysts as well. Now, why is all this significant? This is a very important idea. It's this concept of alternative RNA splicing. If we look up at this image here, we see that, okay, we have these introns in white and then these exons in these lovely colors. Now, during RNA editing, we're going to cut out the introns and then splice together the exons in different ways. But if you notice, using the same segment of DNA, we could splice together a number of different mature messenger RNA segments from the same information. It all depends on how the editing gets done. Think of a long video segment, and you can edit down different portions of the video, and you can come up with, drastically different meanings from the video. That kind of stuff happens all the time. When you look at political interviews and the way meaning in the presentation can be changed by how the video is edited. Well, the type of protein that's made can be changed as well by how this initial transcript is edited. And the resulting finding with this is that you can produce a lot of different proteins with the same number of genes. So you can produce more proteins with a given number of genes than we would have initially anticipated. So, when I was younger, it used to be that people thought, oh, we had 100,000 genes in the human genome. Then it was 60. Then it was 30 to 35. Now we're down to 25,000 genes that are believed to be in the human genome. And there are some people that say that it's actually only a few thousand genes. It could just be like 6,000 or 7,000 genes that can be edited in all these different ways to produce the great variety of proteins that we see. So this is an area of expanding research, and this could be a career path for some people. All right, so let's talk about translation here. Let's see. tRNA is the important molecule at this point. Again, T stands for transfer. By transfer, it implies the fact that these particular RNA molecules carry amino acids to the ribosome so that the amino acids can be linked together by the ribosome to make the protein. Now, you need to be familiar with the structure of the tRNA molecule. Know that one end of it carries a particular amino acid. So if we want to think of it this way, one end has the amino acid. Here's the tRNA. And the other end has a particular anticodon. Now, each particular tRNA is unique. It will have a different anticodon and a different amino acid that it carries. So tRNA molecules are particular to the type of enzyme and type of anticodon it possesses. And here's why. They have a very specific three-dimensional shape, and that's due to the hydrogen bonds here between the bases and the tRNA. So if you sort of flatten out the tRNA, you can see the different regions. And again, because of hydrogen bonding, they'll have this particular three-dimensional structure. What I'd ask you to pay close attention to is what we see here. On the 3' end, this is where the amino acid will attach. So if we want to erase here, let's imagine this is our amino acid. It's going to attach to the 3' end of the tRNA. And at the opposite end, you'll have 3 bases that comprise or make up the anticodon. So if we look, AAG makes up the anticodon for this particular tRNA. So they have a particular shape due to the hydrogen bonding that occurs between the bases. They have a 3' end that sticks out and is the attachment site for amino acids. And they have these anticodons that are complementary to specific codons on the messenger RNA. Here's how it all ties together. tRNAs can float around in the cytoplasm, or inside us all, and they will attach to specific amino acids. They will then transfer or carry these amino acids to the messenger RNA on the ribosome. Now notice how the anticodon is complementary to a particular codon on messenger RNA. That's why it's important that messenger RNA is single-stranded, so that it provides this opportunity for bonding with the anticodons on tRNA. So a particular codon is going to match up with a particular anticodon. And since tRNAs are specific in the amino acid they carry, it's going to impact the order of the amino acids. So here we have this particular tRNA carrying glycine attached. This growing polypeptide is going to link up with this glycine. Once this tRNA has lost its chain, it is discharged and released back. It can pick up another copy of the appropriate amino acid and then eventually work its way back to the ribosome. All right, two parts to this. We'll get into that in just a moment. Let's stop here.